In this video, we're going to talk about arteries, capillaries, and veins. And we'll start with the arteries. The function of an artery, I didn't write it on here, but hopefully you know it. I'll give you a second. If you said carries blood away from the heart, you're correct. So as arteries get further and further from the heart, their structure changes a little bit. So that wall composition can vary in thickness. And basically this is because as we get further and further from the heart, the pressure is dropping in the arteries and we don't need as much muscle in the wall of the artery, so that tunica media shrinks. Also, as we get further and further from the heart, the lumen of the arteries gets smaller and smaller and smaller. The smallest of the arteries are called arterioles, and arterioles are going to deliver blood to capillaries. Arterioles are the smallest of our arteries, and it's the job of the arteriole to deliver blood from the artery to the capillary. The arterioles have branches that lead into capillaries, and the branch that leads into the capillary is often referred to as a metarteriole. So that prefix meta means between. So the metarteriole is basically the blood vessel between the arteriole and the capillary. Now let's take a closer look at capillaries. So as we stated before, capillaries are the main site of exchange between the blood and the tissues of the body. What sorts of things are we exchanging? Nutrients like glucose, amino acids, vitamins and minerals, water, right? Gases like oxygen and carbon dioxide, waste like carbon dioxide or urea. So there's a lot of exchange happening here at the capillaries. In terms of their structure, as we learned previously, the capillary is mostly just made of simple squamous epithelial tissue, which we call endothelium or tunica interna. These simple squamous cells have a special kind of cleft between neighboring cells. So where one cell meets the next, there's a space in between, and that's called an intercellular cleft. So there are these things called tight junctions that are connecting neighboring cells, and that intercellular cleft kind of runs through there. Intercellular clefts are going to be important when we look at capillary exchange in more detail, and that's because fluids like water and dissolved substances can actually travel through the intercellular clefts. The outside of the capillary is coated with a layer of glue essentially to kind of hold it in place. So this is called the extracellular matrix, or sometimes books will call this the basement membrane. So what I want you to do now is to take a look at the picture at the bottom of the screen and see if you can label the items 1 through 7, right? Once you think you have your answer, go ahead and hit play and the answer will be on the next slide. All right, so here's the answer. How'd you do? Number 1 is going to be our simple squamous epithelial cell. Number 2 is supposed to be a red blood cell. Number 3 is a nucleus from a simple squamous epithelial cell. Number 4 is supposed to be a tissue outside of the capillary. A cell outside of the capillary. So this is sort of representing the tissues that we'll be exchanging with at the capillary. Number 5 is what we call interstitial space, or you could label that interstitial fluid because it will be filled with a fluid. Number 6 is representing our intercellular clefts that are between neighboring simple squamous cells of the endothelium. And number 7, that blue line, is representing that basement membrane or extracellular matrix that's gluing the capillary bed in place. Capillaries have a certain flow through them, right? We go from arterial to metarterial to the capillary. And then from there, after the exchange is complete, the blood will flow into what's called a thoroughfare channel, which leads into a venule, which leads into a vein. But capillaries aren't usually just like this one tube. They're found in like these complex networks, and that's called a capillary bed. So in the picture here, we can see a capillary bed. And if you notice where the capillary vessels attach to our metarteriole, you can see there are these straps of muscle tissue. So these are what we call precapillary sphincters. And these are really important to the function of a capillary because this is muscle tissue that can constrict or relax. When that muscle constricts, it's going to cut off blood flow going into that portion of the capillary bed. And when that muscle relaxes or dilates, that means the door is open essentially, and the blood can flow from the met arterial into the capillary bed. By controlling these precapillary sphincters, we can control the blood flow through a capillary bed. So if a tissue is not active, then we don't need the blood flow going through the entire capillary beds of all of that tissue. Like skeletal muscle. I'm not running a marathon right now, so likely my precapillary sphincters are constricted in most of my skeletal muscle because my muscle tissue doesn't need that blood supply. But I'm doing a lot of thinking, so in my brain we're going to have relaxed precapillary sphincters, allowing sufficient blood flow to the capillary beds there. Now let's take a look at the different types of capillaries that are out there. So we have three main types of capillaries that we'll discuss in this class: continuous capillaries, fenestrated capillaries, and sinusoids. Continuous capillaries are the most common, but they're not found in the brain. We have something in the brain called the blood-brain barrier, which is a special type of capillary that has a coating of cells on the outside called astrocytes. We need to be very selective about what sorts of exchange occur between the blood and the brain so that certain chemicals and bacteria, viruses don't get into the brain tissue. The capillary is made of simple squamous epithelial tissue, so endothelium, and it's called continuous because of the basement membrane here. So the basement membrane, or that extracellular matrix, is sealed all across the entire capillary. There's no holes in it. As we'll see with sinusoidal capillaries, there is going to be holes in that basement membrane. Now, just because there's no holes in the basement membrane doesn't mean things can't cross that basement membrane. It's just a layer of basically sticky proteins, so things can still get across. Don't forget we have those intercellular clefts that are between the adjacent epithelial cells, and these clefts are going to allow for the passage of really small molecules through the intercellular clefts. So water, and then small things dissolved in the water, like maybe glucose or gases like carbon dioxide and oxygen. Fenestrated is a term that means opening or window. So fenestrated capillaries have holes in them or windows in them. Because fenestrated capillaries have more holes in them than a continuous capillary, they can allow for more exchange and exchange of larger materials too. When I say larger, I'm not talking like the size of a cell. I'm talking about maybe the size of a protein. So water and small things will get through the fenestrated capillary, and then things like proteins might get through. You're going to find the fenestrated capillaries in areas of the body where we're making protein that needs to be put into the bloodstream. A good example of this is our endocrine glands, which are sometimes making hormones made out of protein. And to get those protein hormones into the bloodstream, we need a larger space, and the fenestrated capillaries come in handy there. The kidneys also have fenestrated capillaries, and there's a lot of exchange that goes on of some larger material in the kidneys. Same thing for the small intestine. There's the exchange of larger materials in the small intestine too. Sinusoidal capillaries are also known as discontinuous capillaries, and they're called discontinuous because the basement membrane of the capillary has holes all over in it. So in addition to this, the simple squamous cells have fenestrations too. So this is like an extreme version of a fenestrated capillary. It's very, 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 very leaky. We're going to see these leaky, discontinuous, or sinusoidal capillaries in areas of the body where we need to move very large materials. And we're going to see this in the bone marrow and the liver and spleen. And if we look at the bone marrow as an example, in the bone marrow, we're making blood cells. So we need to get blood cells out of the bone marrow into the bloodstream. So we need really big holes to do that, to get things like our neutrophils and red blood cells out of the bone marrow into that capillary. We need these really big holes. So those discontinuous capillaries come in really handy there. Take a minute and see if you can guess which picture is representing which type of capillary. Go ahead and pause the video, and when you're ready for the answer, hit play. So if you said this very holy capillary was the sinusoid or discontinuous capillary, you are correct. This is our continuous capillary. Notice that there's no holes at all in the the epithelial cells, and we have a continuous basement membrane. Here's our fenestrated capillary. It has a continuous basement membrane, but there are small holes all throughout the epithelial cells. I want to talk about a couple special topics that apply to capillaries: anastomosis and portal systems. An anastomosis is an alternative pathway for blood flow. And these are really important in the brain and the heart and other areas of the body. And the reason why they're important is because if the path of blood gets blocked flowing to that organ, we have an alternative pathway we can follow. So in the image on the left hand of the screen, we can see our typical capillary bed. But notice over here, we have another way of getting over here. So if we say this is A and this is B, we have two ways to get from A to B, right? So if this becomes clogged, we have this alternative pathway. A portal system is when we have two capillary beds in a row. Typically, it goes arterial, capillary, vein. But sometimes we have arterial, capillary, capillary, vein. And that's what we call a portal system. So we see a portal system in the liver. There's one associated with the hypothalamus and pituitary, and they serve different functions, but I just wanted to mention them. So if you do hear that term portal system, you know what it's describing. If we're following the path of blood flow, next up would be venules, because blood flows from a capillary into a venule. So venules are basically very tiny veins. So take a look at this drawing and see if you can figure out which number would be representing the venules. If you said 4, you are correct, right? Number 1 would be the arteries, 2 would be arterials, 3 would be capillary, 4 is venules, and 5 would be veins. Next up are veins. So veins have two jobs. The first and main job is to return blood to the heart. But veins also serve as a blood reservoir. And the reason why we say that is because at rest, most of our blood is in our veins. So a little over half of our blood is going to be in our veins when we're at rest. And when we need more blood in our arteries to deliver to our capillaries, we can call upon that blood reservoir. So like when we're exercising, we need to have more blood flow going to our skeletal muscles. We're going to relax those precapillary sphincters and get more blood flowing into those capillaries of our skeletal muscle. And so that's a lot of capillary bed coverage, right? We're going to need a lot more blood in our arterial system to deliver to those capillaries. So we can call upon our veins and take more blood out of that blood reservoir. And the consequence of this is that there's going to be less blood in the veins, but we don't really care about that much because really we're just using that to return it back to the heart. How do we return venous blood to the heart if the blood pressure is so low? So there's 4 main things that we're going to use to return blood to the heart. The first one is going to be the fact, okay, there's a tiny bit of blood pressure in there, right? When the heart contracts, it's pushing blood through the arteries, capillaries, into the veins. Additionally, when the heart relaxes, it creates like a suction. So the atria, when they relax, they're like sucking blood into them from the veins, and that suction helps a little bit too. The skeletal muscle pump is really important for venous return to the heart, along with valves. Let's take a look at the picture at the bottom of the screen. We can see skeletal muscles squeezing on a vein here. This is the valve in the vein. Valves are there to ensure one-way flow of blood. So what's going to happen is when this skeletal muscle, or when these skeletal muscles contract, they're going to squeeze on this vein, forcing the blood to go in one direction. The blood can't go down in this picture because of the shape of the cusps of the valve. The blood that did go down would just shut the valve. But here, as the blood goes up, it causes the valve to open. So these valves help to ensure that one-way flow of blood that's always going towards the heart. But we wouldn't be able to do anything with those valves if it wasn't for the skeletal muscles. So that skeletal muscle contraction is really important for venous return to the heart. And the last contributor to venous return to the heart is the respiratory pump. Basically, just by breathing, we're causing changes in pressure in our body, and those changes in pressure are going to squeeze on our veins, and that helps to return the blood to the heart.